Molecular genetic is a subfield of the genetic that focus on the structure and function of gene on a molecular level, including genetic variation, gene expression, and DNA replication and repair. This field aims to understand how genes are transmitted from one generation to the next and how they influence human behavior, health, and disease. Research in molecular genetics rely heavily on laboratory methods and technologies such as DNA sequencing, uh, PCR, and gene editing techniques. Practice or practical application, amplification of DNA sequencing, mapping genes, linkage analysis, diagnose diseases, pathogen screening, sex determination, forensic analysis, paternity and maternity relatedness, behavioral ecology, molecular systematic and evolution, sequencing in different organisms, population genetics, physiological genetics, livestock uh, pedigree, optimized breeding, wildlife management, stock identification and assessment, detection of genetically modified food. Background on the polymerase chain reaction, one of the most important methods in the molecular genetic, ability to generate identical high copy number of DNA made possible in the 1970 by recombinant DNA technology, cloning DNA is time consuming and expensive. This is disadvantage. Proving libraries can be like hunting a needle in a haystack. PCR discovered in 1983 by Kerry Mills enabled the amplification or duplication of millions of copies of DNA sequence with known flanking sequences require only simple inexpensive ingredient and a couple of hours. DNA template and primers anneal to flanking sequence DNA polymerase uh, DNTBS magnesium buffer double-stranded DNA can be performed by hand in a machine called thermocycler. Kerry Mills won the Nobel Prize in Chemistry in 1993 for his discovery. How PCR works begins with DNA containing sequence to be amplified and a pair of systemic oligonucleotide primers that flank the sequence. Next, denature the DNA to single strand at 94 centigrade. Rapidly cool the DNA to 70, 37 to 65 centigrade. Anneal primer to the complementary sequence flanking the target DNA. Extend primer at 70 to 75 centigrade using a heat-resistant DNA polymerase, Taq polymerase, derived from Thermus aquaticus, it's a bacteria. Repeat the cycle of denaturing, annealing, and extension 22 to 45 times to produce 1 million to 35 trillion copies of the target DNA. We amplify the DNA, extend the primer at 70 to 75 once more to allow incomplete extension product in the reaction mixture to extend completely cool at 4 centigrade and store or use amplified PCR product for analysis. How the water bacteria, the Thermus aquaticus, attack DNA polymerase. We get this uh, polymerase from the Thermus aquaticus, the bacteria which live in the hot water. Here the sequence denature to single strand, extend the primer, repeat the denature and annealing, extend the primer, repeat the denature and annealing, extend the primer with Taq DNA polymerase. Here the principal steps: denature, anneal, extend, and repeat. Here the thermal cycler. Example thermal cycler protocol used in lab. Step 17 minutes at 94 initial denature. Step 20 245 cycle 20 second at 94, 20 second and 52, 1 minute at 72 denature and nail extension. Step at 37 minutes at 72 centigrade final extension. Step 4 infinite hold at 4 centigrade storage. Here we can make this in thermal cycler. We put this information in program of the thermal cycler to make these cycles. DNA sequencing, determining the nucleotide sequence of DNA, determining the sequence of, of the nucleotide of the DNA, determine the nucleotide sequence of the DNA developed by Frederick Sanger in 1970, and Walter Gilbert and Frederick Sanger won the Nobel Prize in 1980. Manual DNA sequencing output extension product in each of the 4 reaction mixture, also aimed with different radio label DDD and TB. Next, each reaction mixture is electrophoresed in a separate LAN at high voltage on polyacrylamide gel. Pattern of band in each of the 4 LANs is visualized on X-ray field. Localization of bands in each of the 4 lanes indicate the size of the fragment termination with the respective radio label D, D, and TP. DNA sequence is deduced from the pattern of bands in the 4 lanes. We can see it in animation. It's better to understand. This is the diagram. Radioactive oligonucleotide primer extended by DNA polymerase clone sequence to be analyzed and sequence deduced from the banding pattern and autoradiogram made from the gene. The gel. This is a polyacrylamide gel. Radio labeled DNTPs, 4 runs, short product, long product, sequence 5 to 3. Combine 4 dyes in one reaction tube and electrophore in one LAN on polyacrylamide gel or capillary containing polyacrylamide. UV laser detect dyes and read the sequence. Sequence data is displayed as colored peaks, chromatogram that correspond to the position of each nucleotide in the sequence. Throughput is high, up to 1,200 base per reaction and 96 reaction every 3 hours with capillary sequencer. Most automated DNA sequence can load robotically and operated around the minimal labor. Applied biosystem and applied biosystem. This is the machines we use. Applied biosystem prime, and there are many modern machines. Virtual automated real-time DNA sequence, trace file, and chromatogram for opposite strands. DNA fingerprinting, DNA typing, profiling. Known to individual produced by sexual reproduced organisms, except identical twins, have exactly the same genotypes. Why? Crossing over of chromosome in meiosis phase 1, random alignment of the maternal-paternal chromosome in the meiosis metaphase 1 mutation, DNA replication error, same effect as mutation. This is the causes. There is no two version has the same uh, genetics, DNA fingerprinting, DNA typing, types of marker, restriction site, length polymerase detected by PCR, allele specifically nucleotide probes, repeated DNA, mini satellite, repeated unit of 5 to several 10 pm, discovered by Jeffrey in 1985, microsatellite, short tandem repeated unit of 2 to 6 base pair. This is a marker in fingerprinting. Mini satellite repeat. 4 criteria for selecting useful DNA fingerprinting marker. Marker should be polymorphic so that they are informative. Marker should be single locus so that they occur in only one location in the genome and there is no ambiguity about their number and position. Marker should be neutral so that they are not correlated with selection of adaptation unless selection of the adaptation are to be studied. Marker should be located on different chromosomes so that the markers are independent. Microsatellite short tandem repeat, heterozygote, male, female, homozygote, male, female. One proposed explanation for their fast rate of evolution is slippage during DNA replication. Excellent marker for DNA fingerprinting because polymorphic, single locus, fast evolving, natural, non-coding, common throughout genome of most organisms. How to single-friendly alligate paternity using microsatellite. Extract DNA from the mother, baby, and the alleged father. Synthesize oligonucleotide microsatellite primer and label one primer with fluorescent dye, two primer per microsatellite. Amplify microsatellite using PCR from mother, baby, father. Electrophoresis microsatellite PCR product on DNA sequencer with fluorescent size standard loaded in the same LAN or capillary. 3 to 4 different microsatellites can be multiplexed in each LAN or capillary by using 3 to 4 different fluorescent dye. Calculate size of each microsatellite relative to size standard. This size standard also can be run in the same shell LAN or capillary using 4th or 5th color dye. Sequence at least one copy of each allele to verify alleles. Hypothetical gel pattern for microsatellite heterozygotes for all individuals. Paternity analysis and conclusion. Baby and the mother are expected to share one allele and the baby and father the other allele. If baby and the father don't share a common allele, the father is not the father. No common allele. If the baby and father do share a common allele, paternity is possible. There is a possibility, but not proven. Okay? Not proven because other men in the population also carry the allele at the same frequency. Frequency of alleles that are shared in common or chance can be calculated and an appropriate number of microsatellite analyzed to calculate probability of paternity. To achieve high probability, 60 to 12 loci should be assayed. Exact number depend on variation in population for each marker. If each locus has few alleles, more loci are required. If allelic diversity is high, fewer loci can be analyzed. Single nucleotide polymorphism, DNA sequence of most individuals are most identical, more than 99%. Single base pair difference occur about once every 500 to 1000 base pair. In most population, there is a common SNP and several less common SNPs. About 3 million SNPs occur in human genome, and these are becoming popular genetic markers. SNPs can be used just single nucleotide polymorphism can be used just like other genotyping markers, but more loci typically must be used because only 4 alleles GGCT are possible. How to type single strand, single nucleotide polymorphism? Single nucleotide polymorphism can be typed by hybridizing a complementary oligonucleotide. If the stringency is high, the oligonucleotide will fail to bind to DNA, showing polymorphism. Many hundreds of SNPs can be tested simultaneously using DNA microarray DNA chips, gene chips, SNPs chip, first developed in the early 1990, ordered grade of short complementary no sequence oligonucleotide placed at fixed position on silicon, glass, or nylon substrate. Oligonucleotides are experimentally determined and are either micro-spotted or sensitized on the chip. User-defined SNE chips are available commercially and can contain 400,000 different groups. Oligonucleotide DNA target SNPs single mismatch hybrid is unstable. This match-based pair can't for typing DNA with an oligonucleotide. How to type SNPs? SNPs chip is designed with an array of user-defined oligonucleotide attached to the substrate. Oligonucleotide match each of the common and the variant allele in the population. Target DNA are labeled with a fluorescent tag and hybridized to the chip. Fluorescent pattern is detected by a laser. Because the oligonucleotides are known, the pattern indicates the type of allele the individual possesses. Many different alleles and thousands of different loci can be screened simultaneously in the same experiment. A schematic of the SNP chip assay. Hybrid probe cell labeled CDN target oligonucleotide probe. This is a gene shape probe array of hybridization probe array genetic from the Mendel